Welcome to Chapter 3, Mendelism, the Basic Principles of Inheritance. I'm going to quickly go through the chapter outline and then we'll take each topic. And this talk pretty much follows the information in your text, so be sure to follow along with your textbook. We'll start with Mendel's study of heredity, followed by the application of Mendel's principles, and then testing genetic hypotheses, finishing up with Mendelian principles in human genetics. Now let's just talk a little bit about Gregor Mendel, the often called the father of genetics. He lived from 1822 to 1884, and he published results of genetics experiments on pea plants. He did groundbreaking experiments in 1865 is when they were published. They were those results were ignored for many years. The paper was rediscovered in 1900 by 3 different scientists, and his experimental results were championed by a 4th scientist, and he's the one who coined the term genetics, from the Greek to generate, and that term describes the study of heredity. Now, Gregor Mendel, through his experiments, described many principles and essentially laws of genetics, and many of them hold true today, which is, I think, pretty amazing. So let's talk about his experimental subject, the garden pea. Seemingly a very simple organism, but what made it easy for him, or valuable for him, to conduct his genetics experiments is that this plant has a distinct male and female reproductive organs, the anther and the ovary, respectively. And these organs are contained within the flower, and the petals of this flower close down tightly. And for this reason, self-fertilization is possible, which is the transfer of the male gamete, the sperm, in the pollen grains to the ovary, the female part. And for this reason, many of these strains are inbred, and allowed him to produce true breeding strains. So why is that important? Why do we care about true breeding? Well, true breeding is very important in genetics experiments because if we think about any well-designed scientific experiment, basically a well-designed scientific experiment is only going to have one variable, so just one thing that changes from experimental condition to experimental condition, and that allows us to attribute any changes we see to that one variable. So if we can have true breeding strains, that means that we can study single pea plant traits individually. So he had these true breeding strains for different plant traits. So plants that varied only by height, or only by seed shape, seed color, flower color, pod shape, pod color, or flower position. Those are the 7 traits that he studied. And he had true breeding varieties for each of those traits, and that allowed him to study one variable at a time, and that was very important for his experimental design. All right, so what was his very first experiment? Well, his very first experiment was a monohybrid cross, and mono means one. Hybrid means an organism that has different genetic traits, so monohybrid. And what he did first is he crossed tall and dwarf plants. So here you have one parent plant and the other parent plant, and you can see the different reproductive organs here. Here's the female ovary, and those are the male organs. And what he was able to do is take pollen from one and literally transfer it from one flower to the next. And so in this way, he was able to take tall and dwarf plants, cross them together, and essentially make sure that the offspring he got were only the offspring of these tall and dwarf plants. Now, what he found was that all of these hybrid progeny, so make yourself familiar to the word hybrid, we'll be using that a lot throughout the whole course. All of the, so these are the parents. I think I lost my cursor. There it is. These are the parents. All of the progeny are tall. So what he noticed was that the dwarf trait essentially disappeared. And it didn't matter which way he did this cross. The reciprocal crosses all produced tall progeny, which means that it didn't matter whether he took a dwarf pollen from a dwarf plant and transferred it to the ovary of a tall plant, or vice versa. They all produce tall progeny. Then next thing he did was cross those hybrids together, and I see a little typo on my slide here. That should be an X. This X denotes crossing, or essentially the breeding of these organisms together. I apologize for that little typo. So what he saw when he crossed these all tall progeny together, is he found that he got some tall and some short, or some dwarf. And he got those in about a 3-to-1 ratio every time. So what he concluded was that there was some kind of latent factor. In this case, it was the dwarf, because the dwarf trait sort of disappeared in that first generation. And he called that latent factor recessive. And then he noticed, or he concluded, that there was an expressed factor, which he called dominant. In this case was the tall trait. And the other thing that he realized was that the dominant and recessive factors separated during reproduction, allowing that recessive factor to reappear in the next generation. Now we'll talk a little bit more about that as we continue onward. So he did a lot of monohybrid crosses, again using all 7 of those true breeding strains. And what he found every time was ratios of the phenotypes being the characteristics, the trait that he actually saw in the offspring. He saw them all in a 3-to-1, or approximately 3-to-1 ratio. And he saw what he called the dominant factor in the 3-to-1 ratio, and these are the what we call the F2 progeny, which I haven't defined for you yet. I'll get back to that in a moment. But the parental strains, when he did the tall and the dwarf, he would find all of the F1, that next generation, exhibiting all of the dominant factor. The F2, which is when he crossed those all talls together, you'd get the F2s. So in these F2s, whether he talked about tall and dwarf, round and wrinkled seeds, yellow and green seeds, or any of these other strains, he always saw that 3-to-1 ratio. So what did he conclude from this? Well, he concluded that each trait, whether it was tall or dwarf or seed color or whatever, had 2 forms, and we now call those forms alleles. That term was not coined by Mendel, but that's what we call them now. And he realized that these 2 forms were either dominant or recessive, and that led him to construct his principle of dominance, and we'll talk more about that in a few minutes. Secondly, he concluded that hereditary factors, which we now know are genes, come in pairs, which means that each individual organism has 2 genes. And the way that those individuals get those 2 genes is one from each parent. So he concluded that the parental strains contain these 2 identical copies of each gene. Because they were true breeding, he found that they were homozygous. Not that he used that term, he didn't at the time, but they were homozygous, meaning that the parental strains had 2 identical copies, or 2 identical alleles of each gene. And because they had 2, we now know that means they are diploid. Now, what happens during the formation of the gametes, whether we're talking about the female or the male gametes, is that those alleles are going to segregate, so that each gamete is only going to contain 1 copy, only 1 allele. Now, this is due to the process of meiosis, and that forms these haploid gametes, so gametes that only have 1 allele. And this led him to conclude or to devise his principle of segregation, which we will also get back to in just a few moments. Now, lastly, what he realized was that because these alleles are going to segregate during the formation of the gametes, and so each gamete is going to have only 1 copy, he realized that when those gametes were fused at the time of fertilization, in the hybrid, what you're going to get is that diploid number is going to be restored. You get 1 allele from one parent, 1 from the other, and that gives you a diploid zygote. So the zygote being that 1-celled organism. And if you have 2 different alleles, let's say a dominant and a recessive, you are going to be heterozygous, whereas homozygous is when you'd have either 2 recessive alleles or 2 dominant alleles. So extending from this, he realized that the dominant and recessive alleles coexist in the hybrid, and that in the hybrid, either allele has an equal chance to end up in the gamete. So if you have a heterozygous individual that contains, let's say, a dominant and recessive allele, and that organism makes gametes, those gametes can contain either the dominant or the recessive allele, and there's an equal chance of that happening. And due to random fertilizations, essentially in the wild, not in his controlled environment, with mixed population of gametes, so some gametes containing the dominant and some containing the recessive allele, that would allow a zygote to have two recessive alleles, and then will allow that recessive trait to reappear in that F2 generation. All right, so let's look at this in a symbolic representation here, and this will hopefully make things a little more clear to you for this monohybrid cross. So, as I mentioned earlier, we have these generations. We have the parental generation, which we refer to as P. Here are his two true breeding varieties. And then we have the filial generations, where we abbreviate using an F. So when you cross two parents together, you end up with the F1, the filial one, or the F1 generation. There's the hybrid. When you then cross two F1 generations, or two F1 organisms, individuals, to each other, you end up with the second filial generation, or the F2. There's even a possibility of having an F3 generation, and I'll get back to that in a moment. So what we're seeing in this case is a representation of which alleles these organisms actually have, which alleles you would see in the parental generation, which you would see in the F1, and which you would see in the F2. Now, depending upon which alleles you have, that is going to determine what genotype you have. And in this case, the dominant allele is always written with an uppercase letter, and the recessive allele is always written in the lowercase letter. And again, as I mentioned earlier, is that true breeding strains are always homozygous, whether they're homozygous dominant, homozygous recessive, and that's going to be really important for the rest of the session here. Just keep that in mind. True breeding strains are always homozygous, whereas your hybrids are going to be heterozygous. So in this example, having a dominant allele along with a recessive allele. Your phenotype, another vocabulary word here, is going to be your physical characteristics. So while, for example, here the genotype is heterozygous, the phenotype is tall. Now, if we think about the F3 generation, and we can think about what we would predict for the progeny, the offspring in an F3 generation, let's just go back up here for a moment. So we can predict here, if we have this F1 generation that are heterozygous, we can predict that each of the gametes, a gamete can contain either the dominant allele or the recessive allele. And if you take two heterozygous individuals and cross them together, you're going to get, or self-fertilize essentially, because we know that's possible in these peat plants. We can have a dominant allele is possible in one gamete, or a recessive. So each plant can pass on either a dominant or a recessive. And when you fertilize, let's say, this dominant containing a gamete with this dominant containing gamete, if these fuse, you can end up with a homozygous dominant individual. If this dominant containing gamete fuses with this recessive allele containing gamete, you're going to end up with heterozygous, and the same thing is over here. If, however, you end up with a gamete with a recessive allele fusing with this gamete with a recessive allele, you're going to end up with a homozygous recessive individual. Now, what that means is that your phenotypes in this F2 generation, you're going to end up with 3 talls, even though you only have one dominant homozygous dominant with 2 heterozygous, and you're going to end up with 1 recessive, homozygous recessive individual, and that's going to be a dwarf. And this is how he ended up with his 3-to-1 ratio. Now, when you come down to the F3, this is mentioned in your book, and I'm going to let you do that on your own. Think about what would happen. What would you predict to happen if you self-fertilized a homozygous dominant organism or individual? So capital D, crossed with a capital D. And the same with the, I've lost my cursor again, the same with the heterozygous. What would you find if you cross these together? And what about the homozygous recessive? Now that concludes this portion of Chapter 3. Please join me for Part 2 of Chapter 3, and we'll continue talking about the crosses and the experiments done by Mendel.